bed. Everywhere you look, there's a heart, a hand to hold on to. Everywhere you look, there's a love.
And on today's episode, we're going to be talking about meiosis. In meiosis, we undergo a reduction division, meaning we start with 46 chromosomes and end with gametes containing only 23. In interphase, we have a growing cell that undergoes a DNA replicating step, similar to mitosis. The only difference is the starting number of chromosomes, which in this case is 46. Meiosis begins with prophase 1, where homologous chromosomes pair and form synapses, a unique step to meiosis. The paired chromosomes are called tetrads. The first mechanism of genetic variation in meiosis results from the formation of chiasmata, which joins homologous chromosomes together in the tetrad. Genetic recombination is caused by the crossing over of different chromosomal segments between sister chromatids of opposite homologous chromosomes. Each tetrad contains 2 chromosomes and 4 chromatids, with one of the chromosomes originating from each parent. Prophase 1 is the most complex stage of meiosis and involves 5 substages. In leptotene, each chromosome becomes visible as 2 distinct pieces. In zygotene, homologous chromosomes begin to pair. In pachytene, the sister chromatids at each pair of the homologous chromosomes undergo genetic recombination via crossing over, exchanging segments of genetic information. In diplotene, the paired chromosomes begin to separate into 2 pairs of chromatids. In dikinesis, the separation of homologous chromosomes is complete and crossing over has finished. In prometaphase 1, the nuclear membrane disappears. One kinetochore is formed for every chromosome rather than every chromatid, as in mitosis, and these chromosomes attach to spindle fibers to begin movement into metaphase 1. In metaphase 1, the tetrads comprised of 2 chromosomes align at the metaphase plate of the cell. The orientation is random, resulting in a 50% chance for the daughter cells to either receive the maternal or the paternal version of the chromosome. Then, in anaphase 1, the tetrads separate into individual chromosomes, each with 2 chromatids, which are pulled by the spindle fibers towards opposite poles of the cell. The second mechanism for genetic variation is caused by the random independent assortment of chromosomes into each daughter cell. In telophase 1, the chromosomes have been pulled to opposite poles of the cell. The nuclear membrane may then reform. In cytokinesis 1, the 2 new haploid daughters, each with 23 chromosomes, are pulled apart and begin the next stage of meiosis, meiosis 2.
In prophase 2, the chromatin in the nucleus begin to condense and become visible under the microscope as they appear as individual chromosomes. Each chromosome possesses 2 sister chromatids attached by a centromere. Centrioles begin migrating to opposite ends of the cell and extend their microtubule fibers. The mitotic spindle is formed. Then in prometaphase 2, the nuclear membrane disintegrates. Proteins bind to the centromeres of the chromosomes, creating kinetochores. The spindle fibers from the centrioles attach to the kinetochores and begin to align chromosomes. In metaphase 2, chromosomes line up on the metaphase plate. This organization on the metaphase plate ensures that each new daughter cell will receive 1 sister chromatid from each chromosome. In anaphase 2, the paired chromosomes are separated at the kinetochore into sister chromatids, which are pulled by the spindle fibers towards opposite poles of the cell. In telophase 2, the chromatids arrive at opposite poles. The nuclear membrane is reformed, chromatids relax again and take on the form of chromatin. Then in cytokinesis 2, a total of 4 new genetically different gametes are eventually created, and this concludes the process of meiosis cell division and gamete formation.